From the Oregon State University's Extension Service, you are listening to In the Woods with the Forestry and Natural Resources Program. This podcast brings the forest to listeners by sharing the stories and voices of forest scientists, land managers, and enthusiastic members of the public. Each episode, we will bring you research and science-based information that aims to offer some insight into what we know and are still learning about forest science and management. Stick around to discover a new topic related to forests on each episode.
I'm Scott Leavengood. I'm with the Forestry and Natural Resources Extension Program at OSU. And one of the things we do is this In the Woods podcast. And we have the pleasure this episode of having a chance to talk with Dr. Steve Strauss, who is one of my colleagues in the College of Forestry, and he's a University Distinguished Professor of Forest Biotechnology. Steve, tell us a bit about your background and the primary areas of focus of your research.
Yeah, thank you, Scott. Thanks for having me here. Much appreciate it. So I grew up in Brooklyn, New York, so I'm a city boy. And over time, I got really interested in nature and trees and started doing some backpacking. And that led me to ecology, and that led me to forestry. So that's how I got here. And then over time, I got interested in the field of genetics because the science was exploding with possibility and was interesting. And then people started growing hybrid poplars in Oregon, and they happened to be really amenable to the genetic engineering, biotech techniques that were emerging at the time. So it was an interesting challenge to try to bring this genetics to sort of make this industry even better, industry that was just starting up.
Interesting. What initially inspired you to get into this area of research?
Yeah, the inspiration was definitely, I wanted to save the world. So like a lot of people in my generation, and probably yours, huge environmental problems facing us. And the thinking was that we need science to tell us what to do, how to do it better, how to do it more efficiently. And so that was my motivation, is to try to contribute to producing wood, producing trees in a more environmentally sound manner.
Sweet. If you don't mind me asking one more question, how long have you been doing this type of research?
This is my 40th year at Oregon
really?
State, and I got my PhD in 1985. So I've been at this a while.
Are there historical examples of how humans have modified plants before modern biotechnology? How do you describe to people the mechanics of genetic modification?
I have a class that I've taught for many, many years here about biotech and society and the interactions thereof, the scientific potential, the social issues, acceptance, and how it can have value. Prior to the modern era, where we're doing what we're calling genetic engineering, we modified DNA radically, but we didn't pull it out of the cell and study it and sequence it and maybe rearrange it and put it back in. We modified it in the cell by selecting unusual mutant plants that occurred in nature, or we made hybrids between species on different continents to try to produce lots of diversity and changes in the DNA. But it's happening inside the plant. In this era, we have the scientific technology to pull the DNA out and these days rather efficiently sequence it. We can determine your genome for a few $100s in basically a few hours. And that was, of course, unthinkable 50 years ago. Now we can see how the natural DNA of wild or domesticated plants could be modified, but it might take a long time through conventional breeding. Or if we want to move one gene from one species to another for some reason to enhance nutrition, give it other properties, that we could do a horizontal gene transfer. That would be essentially impossible with conventional breeding. Once you have DNA you want to put into a plant, you have to somehow get it in a cell, and then that cell that's modified in some way has to turn back into a plant, into an organism. And those are two problems. They're a set of separate problems. So we use Agrobacterium to put it in because it's a natural plant genetic engineer. And then we usually use things like plant hormones to get those modified cells to regenerate, to divide and start to produce a multicellular organism. And so there's a lot of plant physiology that goes into how you do that latter step. And there's also a lot of different kinds of strains of Agrobacterium and ways to use it and modify them and modify the genetic engineer itself. In fact, that's a lot of what we do in my laboratory these days, is find better strains of Agrobacterium, figure out how they work, and modify them for higher efficiency.
How does Agrobacterium modify plant DNA, and how can this modification be considered natural, in your opinion?
So like I was starting to say, Agrobacterium is a natural genetic engineer. There's no question about that. It's in nature, and it does its living by putting its DNA into a plant and getting it to turn into a home for the bacterium. So it is natural. And in fact, now we sequence genomes of everything, right? So we have the genomes of thousands and thousands of plants. And when you look at it on a computer, you see Agrobacterium has actually inserted DNA into plants millions of years ago that became part of their genome that just evolved with the plants. So it's really been around. It's really been doing this thing for a very, very long time. And so what we're doing is taking this natural process, and it does it very efficiently because it's evolved to do it efficiently.
Yeah.
So we take that and we tweak the gene so it doesn't make galls anymore. We don't want a home for the bacteria. We just want to use it to transfer genes in so we can get genes in for whatever trade or scientific purposes and have a healthy, normal plant. The other ways of doing this are more physical, like you literally can bombard DNA in. There's a thing called the gene gun. So whenever I tell people about this, you aren't serious, are you? You're shooting? No, we're serious. People sometimes shoot DNA into plants, into cells. You have sort of micro shrapnel that's coated with DNA, and you literally shoot it in at high speed, penetrates a few cell layers deep, and the DNA dissolves off, and some low rate, it goes into plant genomes and becomes a part. So people don't use that much because it's so brute force. So Agrobacterium is much more efficient, and when genes come in, they tend to be much more intact, not pieces of DNA shrapnel. So that's the preferred way to do it all over the world, every crop.
And I would assume using Agrobacterium, it's far more predictable than like the gene gun.
It is, yeah. You grow it and culture it in a certain way. You induce the gene transfer. We know what chemicals. So an Agrobacterium in the environment senses wounded plants. They can only transfer DNA when there's a little micro wound. So it senses that by the chemicals plants exude when they're wounded. So genetic engineers put that chemical right in the medium. We don't wait for the wound. And so that turns the bacterium on, literally, starts transferring its DNA. So it's much more efficient. People started to understand Agrobacterium better in terms of how its genetic mechanisms worked. We're talking about the 60s and 70s, when we really understood it enough to sort of manipulate it and use it to our benefit. And then the first genetically engineered plant ever in the world was 1984, 85. So that's about the time frame, mid 1980s for that.
Okay, interesting. Maybe from more of a context of climate change, do you think misconceptions affect the ability of society to use genetic engineering technology to help plants adapt to rapid climate change?
There's no question about it. It's really a struggle to think about how you could use these techniques of genetic modification in a timely manner, or to be able to use them where you might want to develop, for example, a emerald ash borer resistant ash for Oregon, which is a public interest thing. There is not the money to overcome the regulatory hurdles in a timely manner. There's not the money to invest in the research in a timely manner. It's almost irrelevant. So my feeling is that regulatory gridlock are so great. They've taken this very powerful science and technology and made it almost irrelevant. And if you can produce a heat-tolerant tree that might survive a heat wave, whereas non-modified trees would, and you could show it to them, I think they're going to say, cool, let's do that. So I think if we can get products out that show the benefits, I think people will accept it.
I think, yeah, Steve, you've already addressed this in some ways, more about how plant transformation techniques might help address climate change-related impacts. For agriculture and forestry, you gave the one example of a tree species that could be resistant to, say, our next heat dome. Are there other examples you can think of how these techniques might help, again, address climate change-related impacts?
Yeah, one thing climate change does is it tends to change and promote pests and pathogens. And we know a lot about the genes that are involved in resistance to different pests and pathogens. So that's not directly temperature, but that's something that deals with a consequence of climate change, spreading pest populations.
Say drought and stress, huh?
Drought and stress. Drought and heat stress are more complicated traits. Often pest resistance has a simpler genetic basis. In biotech is a powerful method, but it's not a replacement for conventional breeding. You know, where you have so many different, you have about 30,000 genes in a genome, and genetic engineering tends to work with a half dozen. So you need the kind of conventional breeding foundation. So it's some combination that will be most useful, putting those together. And as I mentioned, the problem is that one of them is highly controlled by the government and these market forces, and the other one is really free to operate. When you make a new hybrid,
Conventional techniques.
you make a new hybrid, you just plant it in the world, and you don't got to file any reports or get permission or get sued by Greenpeace or anything like that.
The last question that I have here is, where do you see the greatest opportunities and, of course, the greatest challenges for your field of research in the next, say, 5 to 10 years? We've got the funding, we've got the regulations. Is there something else you haven't touched on?
One of the challenges is, I mentioned earlier, doing genetic engineering requires putting genes into cells and getting those cells to regenerate. And it turns out in trees, actually in many crops, but trees are probably the worst, that's really hard. So what you're essentially doing is taking like a cell from a leaf and convincing it should start making roots and shoots and become a new tree again. And it thought it was going to be a leaf for the rest of its life till it went to the leaf litter and died. So you're trying to reprogram it. And that's really hard to do. And the fact that we can do it at all is a small miracle. That's something that's incredibly hard to do in animals, but it's also very hard in plants. And the biology of what happens during regeneration and how to kind of manipulate it so it works efficiently is something we work on in a lot of labs, trying to solve that problem. So that's a big challenge, to understand it and then to tweak it. Right now, if you told me, Steve, I've got $1 million, go make an emerald ash borer resistant ash, I'd probably have to spend more than $1 million figuring out how to do the gene transfer and the regeneration, because we don't really know how to do it and we don't understand enough of the science of it. So I'd probably even argue, Scott, I'll work on a different species that's faster, and then we'll come back to ash in 5 years. So I think there's a lot of science related to regeneration in particular, also agrobacterium and gene transfer. That's a big challenge, but I think we have techniques that are effective and we can make progress. Because of politics, we've made it really hard to move forward. So how you change that, how do you change these laws, is really, really difficult.
Anything else that you wanted to ask, Tegan?
My final question usually is just, if you were to leave the viewers, the listeners, with anything from what we discussed today, or given your research and you want the world to have something to think about, what would that be?
I guess the first thing is, I'm a scientist. I've signed on to this because I believe it's the best way of knowing the world, free of your biases and ideologies.
Sure.
So I think we need to invest in science and keep doing it. And it's also not something you can just turn on and off. If you stop investing in science for the next 5 years, you're not going to have trained people who can really do it.
Yeah.
A lot of people already are leaving the field because of what's happening at federal agencies and with grants. So you've got to keep investing in science and investing in the workforce. The other thing is we have to figure out how to get out of our own way better, which I've talked about several times. So that's a real challenge. And in a paper I wrote last year, we need sort of biological innovation and we need social innovation.
Yeah.
We need to do things smarter and faster and more science-based. And we haven't quite come together to figure that out. And so I hope we can do that. Otherwise, all the great science won't get a chance to do much.
And with that, another very brief question is, I think public distrust in the scientific community is really high right now.
It's been growing lately.
How would you suggest moving forward as far as like science communication goes? Does it take like someone like me who's not super familiar with the science, but is communicating and helping people like you communicate these things to the world? Or do you think that scientists have the power to communicate these things themselves and really spread that information?
I think in the world we're in, I think scientists need to be dedicated to communicating with the public. And that's the National Science Foundation. Whenever they give you a grant, they have a thing called Broader Impacts, where you need to bring students into your lab. You need to go talk at the garden club. So that's already kind of wired into the system that scientists need to get out there. But there's so much out there now. You need to be smart about it, kind of like you guys are doing and we're trying to do in our NSF-funded project. I think scientists have to be there, but they really need a team.
Yeah.
And we need to invest in it so it's out there. I think people still, when you look at ratings of credibility, scientists are still high. It's come down over the years, but
it's not
compared to others,
Yeah.
compared to others, people need science. They do, and they know that they do. And we have to keep earning that. Unfortunately, Tegan, our generation is leaving your generation with some huge challenges. All I can say is I'm sorry and good luck.
Well, thank you. I think there are a lot of younger people, younger scientists who want to get into this field and are probably very interested but unsure. What advice do you have to them?
I'd just say if you see something that excites you in the news or in a Google search, don't hesitate to contact the scientist at a lab and say, hey, how do I get involved? We've trained hundreds of students who come in our laboratory and learn, and it's set up a career for many of them. So don't hesitate to get involved.
Well, thank you for that perspective.
Yeah. So for the In the Woods podcast, we always wrap things up with a few questions in what we call a lightning round. And the first question is always, what is your favorite tree species?
Well, it's got to be poplars.
It's got to be poplar.
So that's like cottonwoods and aspens. They're so beautiful, and they're such great scientific subjects. Easy.
Easy. I thought it might be. And then the second question, what's a unique tool that you use that helps you with your work?
You know, the answer there has to be CRISPR.
Okay.
This is this thing that allows you to change any specific gene in the genome with pretty much 100% efficiency.
Wow.
It's just extraordinary. And it works everywhere. It works in every organism. It's very, very broad. So thank you, CRISPR.
Okay. Lastly, what other resources can you share for a listener that would like to learn more about your research?
The best thing would be we have a good website at Oregon State. You can just Google Steve Strauss, Oregon State University. We have all the presentations my students, my postdocs, myself have given, and you can just see what we're doing and what we're saying, including some of the radical things about changing some of the laws to allow innovation to go forward. It's all written down, all in a bunch of very accessible PowerPoints. And you'll also learn about our values and our principles we operate by. It's all on our nice Oregon State website, so have a look.
Excellent. Yes.
Yeah.
Steve, thanks so much for spending some time with us today and for sharing your knowledge with our listeners.
Scott, thank you very much. Tegan, thank you. Really enjoyed the conversation, and go Beavs.
Go Beavs. Yeah. Sweet. Cut.
The In the Woods podcast is produced by Lauren Grand, Jacob Putney, and Scott Leavengood, who are all members of the Oregon State University Forestry and Natural Resources Extension team. Other members of the team who've been involved in the podcast include Carrie Berger, Jason O'Brien, and Stephen Fitzgerald. Episodes are edited and produced by Carrie Cantrell. Music for In the Woods was composed by Jeffrey Heino, and graphic design was created by Christina Freihoff. Funding for In the Woods is provided by Oregon State University Forestry and Natural Resources Extension, the Oregon Forest Resources Institute, and the USDA Renewable Resources Extension Act funding. We hope you enjoyed the episode, and we can't wait to talk to you again next month. Until then, what's in your woods?